one new handout for today, and we'll need our influenza replication cycle that was given out earlier in the week for the discussion. With regard to the midterms are coming back in section this week. So yesterday, obviously, you got your midterm if you had section, or you will today. Occasionally there are grades, clerical errors, and point totals, etc. Or if you have questions on grading, please get those back to me by next Friday after class is the deadline. And so if it's simply a clerical error, just know. But if you think you were shortchanged on points, then write out why you think your answer is deserving of more points. And there's a key on the website that you can refer to on Gaucho Space. I've had a couple of people ask about, what's a numerical grade translate to in terms of a grade? And for a midterm, it's just one step in the whole quarter. But what I do is I throw out the top score, because someone always has me wired. And then I take the next 5% of the papers and figure out what that average is. And so, that takes into account if I give a poor question or whatever. And so then 90% of that value is an A, 80% a B, 60 a C, 40% D, or if it's less than 40, come around and see me. So that's what we do, and here's what the distribution was. So the low was like 48, as I recall, the high was 98, and the average was 79. And so that's kind of how it fell for numbers of people. All right, are there any questions on sort of the process or the midterm at this point? I'll say the reason for having a deadline of next week and after next Friday, if there are questions about regrading, I won't consider them. Because the objective is I want you to look at your midterm relatively quickly after getting it back. Yeah?
Do you do plus and minus scores?
At the end of the quarter, yeah. Right. Plus and minus, like
A minus.
A, B, yeah, right. Sure. Okay, any other questions? All right, so if there are none, where we left off is in our discussion of measles and epidemiology. We didn't quite finish that discussion, but where we left off was that in the Americas, measles has been declared as eliminated, but we're not quite there. And the question is there a bit of a resurgence in the U.S.? And I showed you this distribution plot for the decade 2000 to actually 2013. And in general, there had been less than 100 cases, but there was an occasional spike like 2011 where there are a couple hundred cases. And what's shown here is a breakdown of after the virus is isolated and identified by nucleotide, you can tell where it came from, and what's plotted is the percent of the cases that had been imported, i.e. the individual infected out of the country and then entered into the country. And so, roughly a third to 2/3 of the cases have been in that situation. But then in 2014, there were over 600 cases, 600 to 700 cases, in part because of the Disneyland outbreak that led to change in law in California. So that's what the situation is in the U.S. And what is it in the world, globally? And so globally, measles remains a major problem in that there are roughly 10 million plus cases per year. So substantial globally. And over the last decade, in the order of 100 to 200,000 deaths per year as a result of measles infection. So has a substantial impact. And measles remains a leading cause of death globally in young children. And it's very unfortunate because the vaccine is a very effective vaccine. And so, how does it, what's the cause of death? So it's the immunosuppression that results in the infection, and that immunosuppression in the measles virus infection then turns out to be a major cause of the morbidity. And what we mean by morbidity as a term is simply illness or disease occurrence, is what the term morbidity means, and mortality, which is substantial, 100 to 200,000 cases. And in 2014, it was 115,000 deaths globally attributed to measles. So anyway, mortality, death. So it's a major problem. In addition to the immunosuppressive effects that, of the viral infection, there's a rare neurologic response. And in order of 1 out of 300,000 infections, what's observed is the virus can go persistent. In the nervous system, in the brain. It's almost always fatal when that happens. And that disease is called SSPE, which stands for subacute sclerosing panencephalitis. SSPE. And so it's a rare consequence of measles infection where the virus, as I say, does go persistent in the brain. Okay, so that will conclude our discussion on measles. On measles, well, let's switch gears now to the other family of Myxoviridae. So among the Myxoviridae, we have paramyxoviruses, of which paramyxoviridae, of which measles is our prototype virus. Man, they put a new AV control system in here, and it's challenging. Yeah, so at any rate, paramyxoviruses, and our prototype was measles that we discussed. The other family of myxoviruses are the so-called orthomyxoviridae. And here our prototype is going to be influenza virus. and it's going to be an Influenza A virus. So these two families have some characteristics that are shared. In both cases, for both ortho and paramyxoviruses, viruses, the genome is going to be negative sense single-stranded RNA. But what are some of the characteristics then that are shared, and what's unique for orthomyxoviruses compared to paramyxoviruses? So, among the characteristics, in terms of the virions, the virions in both cases are spherical. The size differs, though, in that influenza and orthomyxoviruses are smaller. They're 80 to 120 nanometers in size, compared to paramyxoviruses, depending upon which virus, is in the order of 100 to 300 nanometers in size. They're enveloped. Both ortho and paramyxoviruses are enveloped. They both have a helical nucleocapsid structure. They both have a virion associated polymerase. So we're going to deliver the negative sense RNA into the cell, but coming in with the particle will be the polymerase that will make the message. And, you know, as I've said, that the RNA genome is negative sense, single-stranded RNA that's not infectious as you isolate the pure nucleic acid. So those are shared characteristics between the two families. Now, what's unique? The first point will be the location of the nucleocapsid that comes following uncoding of our influenza virion, partial uncoding, in that the nucleocapsid in the case of influenza virus will be targeted to the nucleus of the infected cell. Whereas with measles and paramyxoviruses and all the other RNA viruses, all the other viruses we've considered so far, you know, the events are completely cytoplasmic. But for flu and orthomyxoviruses, the nucleus of the infected host is going to be a very important site. And consequently, the sensitivity to actinomycin D is different. Paramyxoviruses like rhabdoviruses and polio are resistant. There's no requirement for cellular host DNA-mediated RNA synthesis. But for influenza, it was discovered early on that influenza, even though it's an RNA virus, it's sensitive to actinomycin D in the laboratory setting. And so that suggested that there were nuclear events going on, and you'll see what they are. So influenza is sensitive to actinomycin D. And then, of course, the arrangement of the minus genome is different in that the genome for paramyxoviruses is monopartite, one molecular species. But the genome for influenza, as I tried to depict in the schematic diagram that I generated for you, is segmented. So we have 8 different RNAs that constitute the genome of influenza viruses. And so that has a very important consequence that you'll appreciate when we get to disease, because of the segmentation, the rate of genetic reassortment, or recombination, if you will, is high, because you have multiple genomic pieces of nucleic acid. Whereas the rate of reassortment for paramyxoviruses and viruses that have a single molecular species is very low, essentially nil, other than the errors of the polymerase. So there's that difference that occurs between the viruses. All right, so fundamental difference, rate of genetic reassortment will be quite high for the orthomyxoviruses, basically nil for paramyxoviruses. We have nuclear events, and hence the orthomyxoviruses are sensitive in the laboratory to actinomycin D, whereas the paramyxoviruses are resistant. All right, so how do we characterize these viruses, orthomyxoviruses? And so they're broken up, influenza viruses, into three groups that are designated A, B, and C, and are characterized antigenetically. And so on your handout from Wednesday, on the back side is a schematic of an influenza virus virion. And so our characterization of influenza viruses into three groups, the groups are A, B, and C, is by type-specific antigens. And the type-specific antigens classically have been the NP protein, i.e. the nucleoprotein, and the M protein, i.e. the matrix protein. And then the influenza viruses are classified into subtypes. And this is based on the surface antigen. And there are two surface antigens that are the envelope glycoproteins that are the viral gene products, the so-called hemagglutinin or HA, and neuraminidase or NA. So for the HA gene product, there are 16 different antigenic forms that are agreed upon that we're going to return to in detail when we get to disease, because the HA is going to be a major neutralization target for neutralizing antibodies. For the NA glycoprotein, there are 9 different subtypes, serotypes. So in nomenclature for these viruses, the nomenclature might go something like A, Hong Kong, 68, H3, and 2. So this would be an influenza A virus isolated in Hong Kong in 1968, and the subtype serology is H3 and 2 for the 2 glycoprotein subtypes. Or the virus A, California, 7, 2009, H1N1. This is the virus that was isolated in the outbreak that started in San Diego in 2009. That was an H1N1 subtype for the 2 glycoproteins. And that got everyone's attention because I have a slide, but with the new system, how do you advance the Well, the next slide was showing the image of a policeman in 1918 with a mask, because the influenza outbreak in 1918 that was quite virulent. There were more deaths from influenza that year than there were from World War I. And the virus in the 1918 pandemic was an H1N1 virus. And so when this was identified as an H1N1 virus, it got everyone's attention. As it turned out, this isolate is much less virulent than this, which you're considering in section this week of paper in terms of the gene function. Right. So this is our nomenclature for influenza viruses, and our prototype virus is going to be an Influenza A virus in terms of our discussion of how influenza viruses multiply and the basis of their pathogenicity. So in terms of multiplication of influenza, and our prototype virus will be an Influenza A virus. We'll come back to the point in terms of the types A, B, and C. The A viruses are a disease problem because the reservoir is not limited to the human. Other animals are a reservoir, including avian species. Whereas B was viewed for some time to be less problematic because it was solely a human virus. But our vaccine does include now 2 different Bs and 2 different As. C is relatively rare. All right, so let's consider the steps of replication of the virus, how influenza multiplies. There are many unique characteristics to this virus, and then causes disease. And so in terms of rate of multiplication on the summary curve that I gave you earlier, so influenza is here, orthomyxoviruses and paramyxoviruses replicate at about a similar rate, and it's slower than picornaviruses and rhabdoviruses, and to somewhat lower yields per cell. All right, so in terms of then the multiplication steps. First step, attachment. And the attachment is through receptor interaction, like all of our other viruses. So on the virus side, the receptor is the surface glycoprotein HA. And it will be the HA1 fragment that's going to be involved in an attachment. On the cell side, it's sialic acid. So this is the first example where it's a carbohydrate structure that the virus uses to attach to the cell. And the sialic acid will have different possible glycosidic bond linkages. And in the human, it's the alpha-2,6 linkage of the sialic acid that's utilized. In terms of the efficiency of attachment, there's polarity, and for orthomyxoviruses, the apical membrane is more efficient than the basolateral membrane when the cells polarized are tested in culture. And so this is just the opposite of what we saw with rhabdoviruses. So we have attachment through HA sialic acid interaction. The next step is penetration. And penetration is by a receptor-mediated endocytosis. And so we bring our envelope virus into the cell in endosomes. The next step will be uncoating, and the uncoating is partial. And as a result of an acidification that occurs mediated by the M2 protein. So the M2 protein, which is a virion protein depicted here in our schematic, is going to be an ion channel. It's a hydrogen ion channel. And so that's what mediates the acidification that occurs, that then facilitates the fusion between the envelope of the virus and the membrane of the endosome, releasing the nucleocapsid. So you have this fusion that occurs. So effectively, M2 protein, which works as a tetramer, is a hydrogen ion channel that facilitates the acidification that you need for the fusion to occur between the envelope and the plasma membrane. And so that fusion, the fusion is mediated by H2. So the H2 subunit, so our glycoprotein HA is the glycoprotein that's synthesized, that will make as a protein in a bit. HA gets cleaved by a cellular protease. And so you have this figure as figure 5 on your handout today. So the HA protein, when it's first synthesized, is designated as HA0. And there's a cleavage that occurs by a cell protease, not a viral protease, but a cellular protease. And that cleavage has to happen in order for our virus that's synthesized to be infectious. So when the cleavage occurs, then the two pieces are called HA1 and HA2. The two fragments are H1 and H2. And these are held together by disulfide bonds. So it's the HA1 portion of the molecule that is important in attachment. It's the sialic acid binding region of the molecule. It's the HA2 C-terminal region of the hemagglutinin that has the fusion activity that mediates the fusion between the envelope and the cell endosome membrane that allows the release of the particle into the cytoplasm. Now this will be the first example that I give to you that's a drug target, because it was discovered that a certain compound called amantadine blocks the ion channel. And if you block the ion channel, then you prevent the acidification and the fusion, and so the nucleocapsid's not released, and you reduce the replication of the virus. So after that was discovered, then various versions of that drug were developed, and there's one that you may have heard of called flumidine. So flumidine, like amantadine, is a molecule that inhibits the M2 ion channel function and inhibits the virus. Some influenza viruses are sensitive to the drug and some are resistant, and we'll return to that point later in the quarter. Okay, so we have acidification and we have release of our nucleocapsid into the cytoplasm.
Question?
Yeah.
Is HA cleaved before stacking or after it's infected new cell?
It's cleaved in the final steps, and it's a cell-dependent protease. Where'd the question come from?
Is it?
And
cleaved
so
before
you
it's
can
stacking?
replicate the virus and get progeny virus that's not infectious. If the cell doesn't have a protease, it cleaves immediately. Okay? Good question. And that's kind of how, part of how all of this was discovered, is that virus was produced and it seemed like normal virus particles, but it wasn't infectious. So there are different cellular proteases, but not all cells have the ability to cleave. All right, so we've got these nucleocapsids. And then what's unique for influenza is they're targeted to the nucleus of the infected cell. And our RNA synthesis events will be nuclear, not cytoplasmic for influenza. So a very unique situation for RNA viruses. Okay, so RNA synthesis. So how does this happen? So the nucleocapsids that are released, the nucleocapsids, which essentially are ribonucleoprotein complexes, they'll have the viral proteins that are the polymerase, and they have our 8 segments of minus single-stranded RNA. And they have the viral protein NP. So the NP viral protein includes what we call an NLS. And what NLS means in, I guess, cell biology, would be the field, is nuclear localization signal. It's what seems to target these nucleocapsids to the nucleus. And so the NP protein has a nuclear localization signal that's important then in the targeting and trafficking of the nucleocapsids to the nucleus of the infected cell. And so then the RNA synthesis occurs, our viral RNA synthesis, and we're going to be in the transcription mode. We're going to first make mRNAs from those 8 minus segments. RNA synthesis occurs within the nucleus of the infected cell. In terms of the localization, the enzymes are viral. It's a virion-associated polymerase. But there will be 3 different proteins that we need that make this polymerase. And what's truly unique is that the primer that allows for the synthesis is cellular RNA. It comes from cellular mRNA. And that's why the virus is sensitive to actinomycin D. Because if you treat with actinomycin D a cell, to review genetics, actinomycin D inhibits DNA-dependent RNA synthesis. So the actinomycin D inhibits cellular mRNA synthesis, and if you inhibit cellular mRNA synthesis, you deplete the pool of primer, so the virus doesn't have a primer to make its own RNA. So the virus is going to basically cannibalize the cell's message to make its own. All right, so how does this happen? So the virion-associated polymerase, this is an advertisement for mantidine hydrochloride, the drug that inhibits flu, and she's taking it and she'll get better. Right? So the virion-associated polymerase is made of 3 viral proteins. And those proteins are PA, PB1, and PB2. So PA is going to be an endonuclease. And on your handout, I gave you a table. You have this table on the handout that lists the 8 gene segments and the proteins that are encoded and the functions of the proteins, like M2, the ion channel. And so for RNA synthesis, there are 3 of the proteins, the largest proteins made of the viral gene products that are required. PA is going to be an endonuclease. PB2 is a cap binding protein. It's going to bind the cellular mRNA's 5' cap. And then PB1 is actually the polymerase that catalyzes the elongation, if you will, using the minus single-stranded RNA template to make our viral transcripts. Times 8. So you can imagine this is happening times 8 because it's happening with each of the genome segments. So you have this as figure 2 of your handout. So we have our virion RNA, our minus single-stranded RNA segment. And so that genome segment in the influenza field is sometimes called vRNA. That's what the V means. It's the segment polarity in the genome. So there are conserved sequences at each end. And these are going to be important for replication. When we transcribe this minus RNA to make the plus copy, which is the mRNA, so that's depicted here. So you'd have the complementary sequence at the 5' end of our plus. So it's A versus U, G, C, etc. That synthesis is primed by a piece of cellular RNA that's 10 to 15 nucleotides long. The figure says 10 to 13. Point is, it's very small. It's very heterogeneous, and this comes from the cell. So this was cannibalized from the cell to then become the primer that allows us to extend off this to make our plus strand. That cellular, this 5' cap, our viral RNA is going to be 5' capped, and there's some sequence. This is actually heterogeneous, and it comes from the cell because that's the primer. And then we get our unique sequence that's the viral sequence. We're in transcription mode, and then there's going to be a U, an oligo U, conceptually similar to what we saw with rhabdovirus and measles. That when, and it's not shown in the schematic, but when the polymerase hits that U stretch, it stutters and then makes the poly A tail. And so this would be times 8, 1 for each of our segments to make our 8 different mRNAs. So again, to recap, the very 5' dozen nucleotides on an influenza message is cellular because it had been stolen, and that's the primer, and that's what I depicted by this little square and squiggly here. This is cellular RNA that ends up being the primer to allow us to prime synthesis of the RNA from the minus strand. And then we don't copy all the way. We hit a U track and we stutter and we polyadenylate, and so we make the poly A tail. So this happens and generates the 8 different mRNAs. And so then mechanistically, you have on your handout figures, you know, if I could get this, we'll get the slide thing figured out with us really soon, I hope. I had slides that depicted the schematic on your figure showing RNA synthesis, but which is a little different from this one. But here again, we have the cellular RNA, and that cellular RNA is bound by PB2, which is the cap binding protein, binds the cellular RNA. And then PB1 actually binds at the 5' region of the virion RNA, and there's a loop in the structure. So the 3' and the 5' ends of our minus RNA essentially are next to each other as a result of this looping. Then what happens is the PA protein, which has the nuclease activity. So this is a subunit, the viral polymerase. That's the nuclease that clips off those dozen cellular RNAs. So that's the nuclease that actually cleaves off this sequence to give a 3' end that the polymerase can extend from. So that's depicted here. So we end up with our cellular RNA heterogeneous sequence, 5' cap. It's going to have a little 3' end. And there's a short region that's complementary. And then the polymerase, the PB1 subunit, then just elongates from that to give us our plus strand RNA. So we have the viral transcription, the mRNA synthesis that occurs. The next step then would be protein synthesis in the infected cell. And so we have these 8 different message transcripts at this point that are synthesized. Then the protein synthesis in our infected cell is cytoplasmic, because the protein synthesis translation is by the host machinery. And so we're going to make a variety of different protein products that will be targeted to different regions, trafficked to different regions in the cell. And so this cytoplasmic protein synthesis, there are various schemes to then allow us to generate the collection of viral proteins from our mRNAs. The first strategy, there are 4 different strategies. The first strategy is the situation where we have one segment, i.e. our minus single-stranded RNA, and it was transcribed by the virion-associated polymerase to make one mRNA, the plus single-stranded RNA, and then this is translated to make one protein. That's the typical sort of strategy where we have 5' cap, 3' poly A, an AUG, and a stop codon. We initiate and we translate and make a protein. And so that happens with gene 1 that makes the PB2 protein, gene segment 3 that makes the PA protein, and gene segment 5 that makes the nucleoprotein. That's the strategy that the virus uses to make those proteins. Then we have the strategy where we have a single segment, a minus single-stranded RNA genome segment that gets transcribed to make an mRNA, and that mRNA gets translated to make one protein, but the protein then goes through proteolytic cleavage, and the cleavage is by a cell protease, as I mentioned. So in this case, the one protein that we make is the hemagglutinin, HA0, that gets cleaved down to give the two fragments, HA1 and HA2, that are held together by disulfide bonds. So we get two subunits made from a precursor protein. The third strategy that influenza uses is the strategy of translating in two open reading frames, a concept that I introduced with measles. And so in this case, in this strategy, we have one segment of minus RNA. We got one mRNA, but from that mRNA, we synthesize two proteins. And the synthesis of the two proteins is the result of translation using two open reading frames in the single messenger RNA. So we have a situation where we have an AUG that's translated to give some protein, stop, and then we would have a different AUG in a different reading frame translated using a different stop to give protein 2. Okay, so this is a strategy that's used for influenza A in gene 2, where we make PB1 that I've introduced to you as a polymerase component, but there's also a product called PB1F2, because it comes from the second reading frame, a different reading frame, and that's going to be a pathogenesis factor in the virus. So we translate in two frames to make two proteins. Then the fourth strategy that influenza uses is the situation where we have one segment, but that segment gives what we call a primary mRNA. It's not the mature mRNA, it's the primary mRNA because it goes through splicing. And this is possible because the transcription is nuclear. And as you know, splicing of cellular RNAs occurs within the nucleus, not the cytoplasm. And in this situation, for one of the genes, two of the genes actually, influenza uses the cellular machinery to splice a primary transcript to then give two different mRNAs, and each of them encode a cognate protein, so you get two proteins. And so, I'll put you off a little bit in terms of the mechanism of splicing, because the splicing was actually discovered with SV40, a different virus, and that's the basis of the Nobel Prize for splicing. We'll go through that with SV40. For now, suffice it, you need to know that the influenza virus is an RNA virus. The transcription is nuclear, and we're using the cell's splicing machinery to generate two mRNAs from one precursor mRNA. And this happens with gene 7 in terms of the proteins that we get. We get M1, which is the main matrix, and the ion channel is the other product from gene 7. It also happens with gene 8 that gives the non-structural protein 1, NS1, and it gives the protein known as NEP. All right, we'll finish our discussion of influenza on Monday and move to aspects of disease and epidemiology. Have a good week. Hang on. So, one more announcement. If for some reason you missed section yesterday and you would like your midterm, which I